From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 36, recorded on December 19th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hi, Vincent. Welcome back.
Thank you. I'm sorry I was out for one episode. I was traveling, but have you gone skiing yet out there?
I have, yeah. It's actually been a good season so far. We've had more snow this year than I think in a couple of decades. Hopefully it'll continue.
Also joining us today from Hong Kong, Tim Chung.
Hi, everyone. Good evening, everyone. It's weird to be doing a podcast at 2 a.m., but here we are. It's also interesting. I might be incoherent. I might just be completely incoherent. We'll see.
That's
how long have you been there?
I've been there for 2 days. I flew in 2 days ago. I actually have no jet lag problem, like instantly. I don't know how. It's 13 hours ahead, and I just seem to be fine. I think being a scientist for so long completely ruined my circadian clock. Yeah, that probably is why.
You may not have jet lag, but there's a little bit of lag, so we have to be patient as we wait for you to answer our questions. But we will
I'll just be very quiet.
Tim, how long is the flight from New York?
Direct flight is probably around 16 hours, maybe 16 hours. But I had to fly via Vancouver because there's so few flights during COVID. So the whole thing took maybe 23 hours, 22 hours.
Wow.
Yeah, got a lot of reading done.
It's a lot of flying, but then again, you're a young guy. You can handle it.
Not so young. Well, rapidly not getting so young. But it was okay. It was okay.
You're younger than I am.
No deep vein thrombosis, hopefully.
Maybe slowly aging might be the better way of saying.
Also joining us from New Orleans, Vivian Morrison.
Hey, everybody. It's been a minute.
It has. It's our last episode of 2022, and we decided to grapple some of the emails that have been accumulating. As Vivian said, if we don't start answering them, people are going to stop sending them. So we're going to try and get through some of them. By the way, if you want to send
that these are coming in from all over the world.
Yes. They're coming in from all over the world, and some of them are quite old. They're months, probably from the beginning of this year.
Yeah.
And so we will make an effort to get through them. And, I'm going to call on you to read them, but anybody can just jump in and put their 2 cents in, okay?
Sounds good.
It looks to me like Tim wants to do the second one, right? So maybe we'll get someone else to do the first one. Maybe Jason, you can go first. How about that with Antonio number 1?
Sure. I'll just read it out then. Antonio writes, great new lessons, thanks and compliments. After retirement, I have time to study and to invade the field of others, which is strictly forbidden in here. I don't know what he means by in here. Presently, I'm looking after glaucoma and the microbiota, including bacteria and viruses. We have some results with 2 ophthalmologists who feel surgery is not yet the last word. Autoantibodies due to antigen mimicry seem to matter a lot in neuromyelitis optica, transverse myelitis, but in glaucoma as well, and multiple sclerosis. Chen et al. And Cell showed that germ-free mice do not develop glaucoma, which it looks like Tim checked out that paper.
It's a cool paper. I can briefly describe it.
Pathogenesis seems to originate from T-cell.
Yeah, because I was going to say, do mice get glaucoma at all? Is this the Alzheimer's thing where it's we have to force them?
You just wait long enough, and they have to. I've got to give them marijuana to reduce the pressure. I don't know. I didn't read enough about this, but I have come across talks where people put little beads surgically put beads inside the eyeballs to elevate the pressure. And when you do that, the optic nerves start dying, so similar to glaucoma. But in this paper, they also increase the pressure of the eye, which led to retinal ganglion cells, so the optic nerve, to start dying. But what they found was that if you remove the gut microbiome, this doesn't happen. And the mechanism is that when you increase the intraocular pressure, so the pressure of the eyeball, the cells start releasing this protein called heat shock proteins inside the eyeball, and then T cells go in and then starts causing havoc. And you can even, once that's happened, you can actually reduce the pressure. You can, I'm guessing, take the beads back out, do something to reduce the pressure. But the glaucoma still progresses because the T cell is still in there, kind of recognizing the heat shock protein. And for whatever reason, the gut microbiome is important. It actually cross-reacts with bacterial heat shock protein. So somehow the gut microbiome having heat shock protein causes the T, I'm guessing the T cells to recognize that. And when that happens in the eye as well, the T cell goes there and start causing glaucoma. So it might be, so both T cells and gut microbiome might be important in glaucoma, which is very unexpected because you think it would just be your eye having physical pressure causing damage and causing the retina. Yeah.
Yeah,
okay.
Interesting. I mean, germ-free mice are pretty screwed up. Their brains are, they don't develop normally at all. And, there's this big question of whether it's because the bacteria are secreting factors that are influencing development directly, or is it through the immune system? And because the germ-free mice obviously don't have normal immune systems either. So yeah, it's been a lot of papers looking at this. All right, so let me finish this message here. Pathogenesis seems to originate from T cells cross-reacting with human HSP, and neurodegeneration ensues. Hence, it's not sufficient to bring intraocular pressure to normal level as autoimmunity continues to proceed. Colleagues in Greece found Helicobacter pylori to be of utmost importance in their local setting. Could you further look into this avenue? All the best with your many endeavors. Have fun with neurologists. And here they are unbearable.
The differentiation.
Thanks again for your help. Yours, Professor Antonio Ponzetto, retired professor of gastroenterology, Department of Medical Science, University of Turin, Italy.
What were you going to say, Vivian?
I was going to say that I don't know if you guys get this, but when I go to a party and people are like, "Oh, what do you do?" I have to start by saying that I'm a scientist and that I study the brain, because as soon as the word neuro comes out of my mouth, they immediately think that I'm a neurologist. And there's a difference between a neuroscientist and a neurologist. And even within neuroscience, just look at the people in this podcast. We all do very different things and look at things from very different perspectives and levels. So I think there may be a couple of other instances of the conflation of neuroscience and neurology
for sure
here.
Especially since
I think
Ori's
it's a
not
really
here.
important distinction. Right. Because some of us don't, some of us chose this profession so we didn't have to interact with humans, and neurologists, you have to really interact with them to see what their issue is. So
it's really funny that this professor in Italy thinks that neurologists are unbearable.
Why? I wonder why that is. Are they just, is it like, if you have a psychologist or psychiatrist as a friend, they're just constantly looking for your problems? Is it like that? Like a neurologist is constantly trying to figure out oh, hey, it looks like you have early signs of fill in the blank.
I thought it was the neurosurgeons that are unbearable.
Yeah.
I think you're That could be. Yeah.
I think those are they
Yeah,
a
I get that
subset of
often too,
neurologists?
when I say I'm a neuroscientist.
Nah.
Oh, okay.
They ask me if I'm a neurologist.
When people
I
ask me
just said
what
I'm a mouse neurologist.
Go ahead, Tim.
So I was going to say, when people ask me what I do for a living, I just say I play with mice, and then they leave me alone.
Yeah, I'm not going to try that one.
When they ask me what I do, I say I work for you.
Oh, I like that.
I work for you.
Because you're NIH funded, or you mean for you, it's like,
NIH funded, your tax dollars, yes.
Mm-hmm.
And I learned that from a woman who's the head of Research America. And she said, tell them you work for them, and then they'll go, huh? And then you got your in to start talking. Because as you said, if you say neuro, then they think that. Or if you say scientist, maybe they'll go away and find somebody else to talk to. But you could have 5 minutes to explain how I work for you. I think that's a great
idea.
I'm definitely going to use that.
Okay, Tim, take that next one, please.
Okay,
From
but
Tim.
I was getting confused because in our notes, Tim's writing Tim.
Oh yeah, sorry. This is not so.
Keep
Everyone
going.
chime in because I'm not. And this is about Alzheimer's disease and CTE. I'm definitely not an expert, but here we go. Tim writes, and this is Tim, that is not me. Vincent, Ori, Jason, awesome show. I looked up the tables of therapeutics Ori mentioned and commented them on the Micro TV blog post and below. And he cites a paper, a review paper by Long and Holtzman on Alzheimer's disease, an update on pathobiology and treatment strategies, published in Cell in 2019. And that is here for other listeners. Outcomes are not good except for statins, which have their own issues. I did not know that statins are good for... you know what? There's a recent... So statins are for treating high cholesterol, and there are recent papers for... there's a recent paper from the Huisai that suggested Alzheimer's patients might have dysregulated cholesterol productions. Anyway, moving on. I learned a ton from the show and read through the paper. Jason's commonality of inflammation, immune and glial involvement is spot on. Sleep is also a symptom and contributor to spread as well. The Wikipedia entry on Alzheimer's disease has a good image of spread in the brain. And Tim continued to say, I played football from second grade, so I'm guessing it's American football. Since we just had the World Cup, probably should clarify. Wait, I should have a quick look. Is Tim from America? Portland, Oregon, yes. So
Sorry,
Tim
no,
played football.
Oh,
Can you guys hear me? Sorry.
yeah.
Yeah.
Oh, sorry. Yeah, that's allowed. I was scrolling down to see where Tim is from. Tim is from Portland, Oregon. I played football from second grade through high school and followed the chronic traumatic encephalopathy work out of my own paranoia. Paranoia is perhaps a symptoms worried face. This image hits back to a similar cartoon I saw in a paper from 2015. And Tim talks about a review on chronic traumatic encephalopathy. And I'll just quickly, I kind of looked up the paper that Tim cited. And so briefly mention what's going on is that chronic traumatic encephalopathy is happen usually to athletes who experience repeated hits on the head. So American footballers, of course, it's a very kind of impact-driven sport. Even though they wear helmets, there's a lot of impacts on the head. Same for actually football, soccer, football, and also like things like boxing and stuff. And the pathology, and what you get is a lot of athletes actually suffer from dementia, and when they die and their brains are examined, they have a lot of pathology there. And one of the pathology that happens is they have this protein called tau that is also shows sign of pathology in Alzheimer's disease. And this protein called tau, when it's associated disease, it exists in a hyperphosphorylated form. So there's a lot of phosphates kind of covalently attached to it. What's interesting is that in Alzheimer's disease, the tau protein actually spreads from the brainstem up towards the cortex as the disease worsens and progresses. But for chronic traumatic encephalopathy, it spreads from the cortex down to the brainstem. So it kind of goes the opposite direction. So Tim says...
And this is actually... the delay is like half a minute almost.
Yeah.
My lab's working on this idea of how tau spreads from cell to cell. And, this is actually a really common theme in neurodegeneration, that there's always a focal point where the pathology seems to start and then it spreads to other parts of the brain. But why certain areas are more vulnerable, why they start, for example, here in the cortex for CTE versus temporal lobe for Alzheimer's is not clear. But the spread seems to happen through connections. So it's not just random, it's through what looks like synaptic connections between those regions. So we're trying to understand what's involved in that spread and at least trying to understand the spread part, not necessarily like why it starts in a certain place. Although for football, of course, it seems to make sense that the cortex would start being more vulnerable because that's the most that's the biggest area that's getting hit. And Alzheimer's, the temporal lobe. So I think he mentions down the road there that tau is sort of tau pathology lags behind A beta and the plaques. And so there is this idea that tau is the tau pathology is initiated by the amyloid A beta. And why that's happening in the hippocampus is not really clear. But it seems to certainly also mirror the cognitive deficits that you see in these different, conditions. And in Alzheimer's, the tau pathology is more correlated with those cognitive deficits in the amyloid. So that's why there's sort of this idea now that you can't just treat Alzheimer's with these amyloid blockers. You need to look at the tau.
Yeah, I just want to make one more point that I have to remind myself of. Even though we draw connections between, or we try to make comparisons and find similarities between Alzheimer's disease pathogenesis and CTE pathogenesis and sometimes stroke, because stroke is often a precursor or can increase the risk of developing dementia. It's worth mentioning that CTE and stroke are both acute in their first part. I mean, certainly if it's continued concussive events, sure, that's like a little more chronic, you could argue, but stroke and TBI are both acute. And so the events that are happening right after the injury are really different than what's occurring in a brain that is eventually going to develop Alzheimer's disease. So I don't know what the answers are to any of the questions that might be raised there, but I think it's important for all of us to remember that we can't compare very easily between these neurodegenerative diseases, even though we place them under that umbrella.
Yeah, no, that's a good point. Although it is interesting that it's the similar pathology that ends up being diagnostic of some of these diseases. And the tau, there's also all these different versions of tau that are mutated in different kinds of neurodegenerative disorders. Tim, looks like you're back. Maybe. Okay,
I'm going to continue. So Tim goes on about how he talks about how cognition is usually, I know my lag is pretty bad. Tim talks about how cognition is actually usually the diagnosis for Alzheimer's and also for CTE as well. And often by the time you have cognitive problems, your brain already is quite severely damaged. Because if you look at for Alzheimer's, you have tau lagging behind A beta plaque, and then you have cognitive deficits lagging behind tau. So it's kind of it's not the best way for people to be diagnosed. It's just very slow and very delayed. And Tim also actually has theory that it potentially. Oh, to mention that it is interesting that A beta and Alzheimer's disease occur exclusively in the cognitive nerves, however, and not in the motor nerves, so motor versus cognitive regions. This points to oligodendrocytes.
Let's talk about that for a second.
Sorry, yeah, please go ahead.
Forgive me, but nerves and neurons are like, there's some terminology here. Like nerves are peripheral nervous system and they're bundles of axons, but the environment of the nerve is very different than what we see in the brain. Certainly in the brain, we do see large tracts of axons moving in the same direction, like the corpus callosum and other white matter tracts that go from the front to the back, but they're not surrounded by layers of connective tissue and fibroblasts and things like that play a huge role. And also in the peripheral nervous system, the myelin is made by Schwann cells, not by oligodendrocytes. So really, I think it's important to recognize, again, it comes back to making these comparisons. It's very hard to say, or to make comparisons between nerves and then axonal pathways in the central nervous system. But I do think he makes, and then also cognitive to me is maybe not the term I would use, but I'm not sure what word I would use for that. But I do think it's interesting that, have you guys seen a lot about A beta and tau in the spinal cord? I did a quick look. I didn't really see much, which is very interesting. So the spinal cord has gray matter and white matter like the brain does. And it's very well segregated. Like you're not going to have myelin showing up in the gray matter. The gray matter is neuronal cell bodies, and the white matter are axon tracts. And those axons are very long, so they need myelin. And myelin is produced by a glial cell type called oligodendrocytes. The oligodendrocyte basically takes a part of its membrane, extends it, and wraps it around the axons. And that covering creates insulation insulates the axon and allows the electrical signals that constitute neurotransmission to travel from point A to point B without being lost on the way. But in addition to that, and actually some people posit that the oligodendrocytes, because they're covering the axons, they're actually preventing the transfer or kind of the movement of energy or resources. They're creating a barrier between the outside world, the extracellular space, and the axon. And so the oligodendrocytes have developed, they now have two roles. One is to supply the neuron with the energy that's required to do its thing, in addition to, insulating the wires of the brain.
Vivian, whether you remember the episode we did about oligodendrocytes feeding axons exosomes to tell it to kick its mitochondria in gear. Case in point. Yeah, exactly. And so, while there is quite a bit of really interesting research out there about, myelin and white matter and Alzheimer's disease and injury and disease in the white matter and how that relates to Alzheimer's disease pathogenesis, whether it precedes the gray matter damage or if it follows or what the relationship is there. I wonder if the fact that there is so much white matter and so many oligodendrocytes in the spinal white cord white matter, that they're
the neurons there are receiving a lot of metabolic support. And this leads me to think about the fact that some people are calling Alzheimer's disease diabetes type 3 because it's possible that certain brain regions or certain brains don't metabolize glucose as well as others. And that maybe the neurons at that time or the glial cells at that time would work better, so to speak, if they were using lipids or fats as their primary source of energy. So I'm wondering if, it's a very interesting question that Tim, the listener, brings up between how white matter might not be as affected, at least in the spinal cord, as in as gray matter or in the brain. And I wonder if it has to do with differences in metabolism based on the environment being very different in those two places, spinal cord versus brain.
Right. I mean, of course, there are diseases that just affect the spinal cord and motor neurons, like ALS. And the idea there is that there's also similar sort of inflammatory responses that kick in with and that affect these toxic proteins like TDP-43. So I think it's a combination of environment, but it's also that these specific proteins have functions in these cells that you're disrupting. And so that's one thing that's sort of glossed over, is that yes, you have these toxic proteins, they eventually kill the cell, but the early manifestation of these diseases is that you start to get synaptic dysfunction and cellular processes are going wrong. And so the cell type specificity is probably just what these proteins are normally doing are more involved in, for example, plasticity mechanisms in CNS versus maintenance of health in the spinal cord.
That's a really good point. You don't have a lot of you don't have much of those much plasticity going on, and in fact, myelin is a way to close the window on plasticity because once it wraps itself around different parts of the neuron, it's going to prevent synapses from forming there. So that's a really interesting point.
Okay, should I continue? I'm going to continue.
Sure.
So Tim continues to write, I would like you to do a paper on the presence of metal in the brain as a possible upstream precursor. I haven't read that material. Jason mentioned this as an alternative hypothesis. I actually don't remember Jason mentioning this.
Yeah.
Well, there's been a lot of
old idea
episodes.
that metal toxicity, especially copper and aluminium, that hypothesis I don't think has really panned out. There's not a lot of epidemiology data or human data to sort of really support it, to be honest.
Okay. Because I know, sorry.
Or maybe Tim's about to say the thing that I was going to say.
Please go ahead, say it.
I was going to say, what about autism? I feel like I heard or maybe that also is just like a red herring.
I think, there's a lot of environmental factors that I think contribute to brain damage. So air pollutants, for example, and metals, certainly high levels of metal like lead in your drinking water or whatnot is detrimental. Mercury is super toxic. So it's clearly these metals, high concentrations of these kind of metals are toxic for the brain. But whether there's sort of this causation with these neurodegenerative diseases that are very common, I think is probably not really clear.
I was going to say for Parkinson's disease, there's been speculations that increases in iron can lead to degenerative of neurons via this process called ferroptosis. Like it even has its own name. So instead of apoptosis and pyroptosis, ferroptosis is when too much iron causes a cell to die. But I don't know, since the world has industrialized, there probably is a lot more metal around us that we ingest and come into contact with than before. So maybe that could explain why there's more neurodegenerative diseases than previously. or we just live so long that
We're
start
living
seeing
a lot
it.
longer.
The major risk factor is aging,
Yeah.
and the fact that we can keep people alive much longer, but they can't keep their brains healthy.
And then, okay, Tim goes on to talk about one last thing about CTE, which is chronic traumatic encephalopathy versus Alzheimer's disease. Top-down and bottom-up spread of CTE and AD beg the reality that the two are often comorbid. This would not be easy to separate out, but the tau and A-beta deposit in CTE patients are different in the sulcus. This is modeled as a stress point similar to a crenulated candy wrapper. I'm guessing that is a bit like how the cortex is folded folded up. I wonder what the model of AD is to start in the brainstem. It seems like certain areas are prone to AD, like the hippocampi. In that same vein, why is the cerebellum seemingly spared? True for all AD, CTE, and CTE patients. I don't have a model for that, so I don't know enough about that. I don't know if you guys want to comment.
Well, yeah. I would say that, and I don't know, you guys tell me what you think, because I'm not yet an expert on this stuff. Also, disclaimer for everyone listening, I think most of us know some stuff, but we really only know a lot about like a few things. So we're just trying to, science out, use our science tools. But so I just was recently looking at some related papers and saw that, actually the cortex, a lot of areas in the brain will start showing these plaques and these tangles. The timing of onset is a little bit different. But I wonder if we tend to think about the hippocampus and, the hippocampus as being the primary target of these things simply because the deficits that it creates, like once that area starts to not function so well, those deficits are like really obvious to us. Versus there might be like, other things that we don't pick up on in our, day-to-day interactions with people that we don't live with, that are due to the aggregation of these plaques, the formation of these plaques, other parts of the brain, like the prefrontal cortex, for example. Like, if you see somebody making poor choices, you tend to just be like, well, that person must have a high risk tolerance, or maybe they just make poor choices, but that your initial thought is not, I wonder if they're starting to develop signs of dementia due to plaque aggregation in their prefrontal cortex.
Yeah, I mean, that's true to some degree, but I think for example, there is a dementia called frontotemporal dementia, where that is the first and most obvious structure that goes. And when if you look up videos of patients with this, or like family members that talk about their experiences with their sick, affected loved ones, it's a very dramatic change in personality. It's not just like, oh yeah, they started to have some really weird quirks. It's literally like one day they are an amazing businessman that's very successful, to the next day that they just sold their house to their to some scam, because they end up really being affected dramatically.
Yeah, and actually,
it's the disease is gradual, so to people it looks like it's a dramatic change. But of course, as you said, I think a lot of those personality traits, you don't notice them unless you know them really well.
Yeah, and I think this discussion that we're having right now relates to a couple of other questions we may or may not have time to get to about adolescence and some other psychiatric diseases like bipolar disorder, ADHD, ADD, and developmental disorders like autism that also impact heavily the prefrontal cortex.
But, going back to this idea that Tim brought up about the cerebellum being spared, I think it's really interesting in that we sort of untapped resource to sort of figure out what other resilient factors, or are there protective mechanisms in these other structures that are not affected, versus just saying, okay, well, these other cell types are more vulnerable. So, but because there are diseases like there's a various ataxia kinds of neurodegenerative diseases that do selectively affect the cerebellum.
And also, I think hypoxia can really damage the cerebellum quite easily. And also drinking, one of the things that really shrinks cerebellum is, alcohol and alcoholism. So I think different causes can cause different brain regions to suffer differently, I guess.
Yeah, and I want to add to that by saying that recently I have become, in the past year, have become very interested in things, what I'll just kind of call, generally speaking, non-neural things impacting neural function. And those non-neural things include extracellular matrix, which is basically kind of a mesh of different kinds of proteins that provide structure. And so like the springiness of a tissue or the rigidity of a tissue is due to the structure of the extracellular matrix and its ability to hold or move water, for example. But it's also a huge factor in delivering growth factors to cells and or keeping certain proteins away from places they shouldn't be, that kind of stuff. So I think it's really, and I think I don't know if it was Tim or somebody else, one of the other listeners who brought up extracellular matrix. And I think that's very interesting. And I did do a little bit of research and found some papers that look at changes in the extracellular matrix architecture and how that influences, or I think some of these were kind of, observation level where you say, hey, well, let's look at let's see whether the extracellular matrix and its composition has changed in the Alzheimer's disease mouse model brain, or looking at postmortem tissue from humans. The other thing that I think maybe that's interesting is looking at vasculature. So I recently, I'm doing some work looking at A beta clearance through vessels. And in attempting to determine which region or we should look at, or if we wanted to look at all of those, I came across a paper that does a whole brain comparison of vascularization and vascular structure in different parts of the brain. And there's a figure that shows massive differences in vascularization between the cerebellum, which is highly vascularized, tons of blood vessels. The cortex also has a ton of blood vessels, so it's slightly less. And then the hippocampus and the striatum have significantly fewer blood vessels. And if blood vessels are a method for clearing A beta and are necessary for reducing plaque load, the lack of blood vessels could be a reason, in the hippocampus, could be a reason why that area is preferentially affected in Alzheimer's disease. So it's a very interesting part of the Alzheimer's disease research field, and it kind of is the confluence of that with people who study vasculature. So I think the listener brings up some really interesting points.
Those are interesting in that there's certain mutations in APP that cause predominantly a vasculature form of Alzheimer's, because the A beta sticks to the blood vessels.
Right.
But it brings up that point that someone else, I think, writes about here, and that's sleep, which is this sort of general emerging idea that during sleep, the CSF, the cerebrospinal fluid, gets pumped in and out in a way to sort of clear out the junk. And so that's pretty cool. Yeah,
just think about the extracellular matrix. Like I said, that changed its ability to open up and allow more fluid to move through it is probably necessary for the phenomenon you were just talking about. Also, I would like to bring up, for those of you who are doing science, you should write down what time you do anything with your mice, what time you euthanize them. What time do you look at their behavior? Because if circadian rhythms are impacting things like this, that is a really important variable, because mice also don't have the same rhythms as we do, right? Being nocturnal and all. Well, there's some humans that are nocturnal, right? But
yeah, mice are nocturnal. But we screw them up. We make them have the lights on during the day, right? And then we make it dark at night.
They have no way of knowing in theory, they have no way of knowing what's happening in the real world. We typically don't have windows.
You don't think they have a cycle that says it's night now, but the lights are on?
They've been in this facility. Some of them are born in the facility and never see the outside.
I suppose.
Although I do wonder.
And we can change them. We can do the reverse cycle. We've done this in our lab, where we put them on the reverse cycle, and they transition to that. And then when they fully transition, then they don't know the difference.
And do you see those differences in biochemistry, like protein levels or things like that? Yeah.
Yep.
Interesting.
Yep.
Very interesting.
A lot of proteins show a circadian expression level difference.
So I speaking of neurologists, I ran into a neurologist at ASV this summer, American Society for Virology, up in Albany, New York, and he takes care of patients with MS. And he said to me, Vincent, the best thing you can do to reduce your risk of Alzheimer's is exercise. So I started doing that. And Tim actually writes, exercise, sleep, and lifelong learning. So I got the exercise down. I got the lifelong learning down. Can't figure out the sleep part yet.
Well, sleep is the hardest part. Like, why? We don't have to do anything. We just have to lay there.
I wake up. I can't sleep. I don't get it.
Melatonin.
Actually, someone said to take high dose or timed release or something. Because, you can take melatonin and fall asleep, and then in 2 hours you wake up again. But apparently there's a timed release. I haven't tried it. But I don't really want to take drugs to sleep. I want to sleep on my own. Anyway.
Yeah, you have to determine why you're not sleeping.
I have some ideas, but, those are, I can't fix them. That's
Is it related to the lifelong learning?
Yeah, it is, actually. There's partly. There are other things as well that are not related to lifelong learning. But anyway, Vivian, can you take the next one? That's Lori.
Okay, let me see. I've written so many notes that I can't even find the questions anymore. Let's see. Okay, Lori writes, hello, TWiN hosts. I am writing to you with a question that is close to my heart and very difficult to put into words. So if you're listening right now and it might be difficult for you or triggering for you to hear about something happening to children or babies, you may just want to skip ahead. Especially if you're a new parent, you probably don't want to be listening to this part. And it's very difficult to put into words what she's experienced. A few years ago, my 18-month-old nephew was found dead in his crib. He had received his MMR vaccine earlier that day. You can imagine the pain that this has caused, and you can also imagine the direction his parents' thoughts went regarding vaccination, especially with the influence of certain groups on the internet. Many months later, the coroner's report stated unknown cause of death, but one of the findings showed that there was some encephalopathy, specifically an enlarged brain. More recently, the family has had 2 more additions to their family, twin girls. Due to the situation with their brother, the medical advice was to forego vaccination until the girls are of school age. At the age of 18 months, one of the girls was diagnosed with autism. She did not receive the vaccine. And at the age of 2 years, the same girl developed seizures. Testing has been done, it's genetic testing, and they discovered a mutation in the SCN1A gene, which is a sodium channel, which sodium channels are expressed. There are many of them, and they're very important for generating action potentials in neurons. And a possible, if she puts in parentheses, possible diagnosis of, and I'm not sure how to say this. I think the person may have been French to begin with, so it would be Dravet syndrome.
It's Dravet.
Yeah,
Dravet
syndrome, yeah.
Oh, Dravet, Dravet.
Yeah, I mean, it's different in English because we put the emphasis on the wrong syllable, right? To me, this rare diagnosis answers some questions about my nephew's death. It might just be that my nephew saved his sister's life. Could you please talk about Dravet syndrome and how getting a vaccination would result in encephalopathy? That's the enlarged brain. Encephalopathy is a general term, so we usually would add additional qualifiers to describe what we're seeing. Is there a treatment, and is it safe for individuals with this syndrome to get vaccinations later in life? So I'd never heard of this syndrome before, so I did have to look it up. But one of the things that jumped out to me was the fact that the seizures that these individuals experience, and by the way, these individuals do not live very long. I think the seizures become intractable. And also you have which happened to Lori's nephew. Essentially, it's like sudden infant death syndrome. And there actually is another acronym that's sudden unexpected death in individuals with Dravet syndrome. So anyway, the seizures are temperature sensitive. Which is actually one of the things that people have thought about with sudden infant death syndrome, that overdressing them or keeping them in a warm room or having their face close to, or, having blankets close to the face, that the temperature is doing something to decrease, to bring breathing down or something like that, which then ultimately leads to death. So bringing it back to that temperature sensitivity, vaccines can cause fevers. And it's very, so it's possible that in some individuals who have this mutation, and there are actually a handful of mutations, quite a few mutations, that can develop into this seizure disorder. And so perhaps if we knew ahead of time, if you have these mutations, you shouldn't not vaccinate your child. That's my opinion. I think that the life, provided they don't die from the vaccine, as in this case, that the lifelong value of being vaccinated is far outweighs the risk in terms of probability of getting the vaccine. But we could, if we included this kind of genetic sequencing in the tests that we have done, as the baby's gestating, you could identify this and then try to just have some system in place or some prophylactic treatment, something that would help prevent the fever from occurring, that would be useful in protecting these children around the time that they get vaccinated. And I just want to remind everyone of the recent experiences they and everybody they know have had with the COVID vaccine. Some people were hit really hard, and some people were not impacted at all. So it's a difficult situation. As I think Vincent once said, the COVID vaccine is, plays by all the rules, but what's really going to come out of this is that we're going to start understanding more about human immunity, and because there's such differences. And so I think, it's, I think that perhaps that like the genotyping prior to the baby being born could be helpful.
Yep. Perfect. Yeah.
Yeah, I have a colleague here who works on Dravet syndrome, and it is very rare but severe epileptic disorder, and of course, as you mentioned, the mortality rate's low anyway. So this is clearly a very rare interaction that some babies have with the vaccine, where it can cause death, and without knowing that there's this mutation, of course, you wouldn't know that this would be a dangerous thing to do. And so I agree. I mean, at some point we're going to have the ability to do whole exome sequencing of everyone when you're born, and the question will be what to do with that information once you get it. And in this case, where there's a clear known mutation that can cause the disorder, obviously, then you can really act on it. But many variants, many mutations and genes are not one-to-one. It's not like if you have that mutation, you will get a disease. But anyway, back to the vaccines. I agree with you. These are very rare events, and for the most part, they're very safe.
I guess Vivian's point is right, that if you know the genetic risk factors, then you do genome sequencing, and then you postpone vaccination, right? Mm-hmm. Till later. That's good. All right, I will take the next one. Tim. Could be the same Tim. Let's see. I don't know. No,
he's got two questions back to back. There's just a lot of Tims.
It might be the same one. TWiN, this show, Worms See the Light. Okay, so that's episode 17 in April 2021, where we had Michael Niddeback on to talk about C. elegans. C. elegans, of course, doesn't have eyes, yet they can discriminate between colors. So this show brings out more questions than answers, which is always good. I've read about C. elegans before, but this show led me to crack the books a bit more to speculate where these light-sensing molecules might occur. The story of this worm's simple connectome is legion in scientific communication of the abstrusity of neuroscience.
There's some GRE words in there for you.
Abstrusity. Wow.
Yeah, that's a new one.
It is often said, if we can't figure this worm out, what hope do we have figuring out our own brain with 85 to 128 billion neurons, depending on how you count them? I think there's more hope with the human brain than this worm, perhaps. After reading The Structure of the Nervous System of the Nematode C. elegans by J.G. White et al. Most of the afternoon, I have to say I'm discouraged from ever understanding the function of the human nervous system based on such simple models. There's nothing simple about the worm. It may only have 302 neurons, but those all fall into 118 structural classes that dictate functional class. Evidently, there are en passant synaptic connections. I'm not sure exactly how those work or if they are conserved in mammals. Are they? Mike was very on point, saying C. elegans might survive without a nervous system, as the pharynx is almost entirely independent, save for two interneurons, though eating isn't everything. Defecation might be more problematic without some neural stimulation.
I definitely laughed out loud at that last bit. So,
yeah, this goes to this idea that neuroscientists are trying to do what's called a connectome map of brain tissue, and the OG is the C. elegans connectome, which is because it's got 300 neurons. And even with the C. elegans connectome, we know the map. We still don't understand exactly how all the neurons function. And that's partly because you may have the map, but you don't know the cars that are transiting on the map, or whether there are cars and trains, or cars and trains and planes.
Like that movie, right? Yeah. Steve Martin.
Well, I don't have any insight. I, from talking to neuroscientists, I get the impression that it's going to be hard to figure out our own brain.
Yeah, when I was, so I have two anecdotes. The first is that when I was in college, I took a class on wild animal models in neuroscience. So looking at, corvids like crows and blue jays and anglerfish, things like that. And I remember having like an epiphany, which was like, we are never going to understand anything. Like it seems impossible. But the other was that made me think about this paper actually returned to it, was that my son said, my 6-year-old son, I picked him up from school one day. We were just pulling out of the carpool line, and he said, how do we see anything? And I was like, what do you mean? And he's like, well, how do we see anything? Like, is the stuff we see really there? And I was like, whoa, like, what is that, Descartes? Like, is this cup really a cup?
Matrix.
And I was like,
we're in the Matrix.
Exactly. I was like, wow, this kid is smart. Just pat
That's very
myself on
good.
the back there.
That's very good.
Yeah, but I definitely understand I feel for Tim, because I personally have been there, being like, how are we going to figure this out? Do we need to figure it out, though?
Well, that's another question entirely, right? Yeah, to a certain extent, if you want to figure out how to cure or prevent diseases, right? That's a lot of what we do. Because, like the baby who died at 18 months, we'd like to prevent that, right? Mm-hmm. So I think to a certain extent, yeah, we should know. But also, it's all about curiosity. I think even if we if we had no diseases, we'd still do science just because we want to know how it works, right?
Mm-hmm. I mean, some of us do science for that reason anyway.
It would be incredibly frustrating. Imagine if we understand everything else about science except for ourselves. That's the one thing we can never understand.
I think it comes down to what understanding means. I don't think any one of us can understand what the human brain is or how it functions. And so I think we need to sort of figure out, well, what aspects are that we need to understand in terms of disease, and that's a solvable problem. Mm-hmm. But it's interesting to see how now artificial intelligence and machine learning is finally getting to the point where it's mimicking performance of brains, but we still don't actually know if that's how the brain works.
Yep.
And so that's where this understanding,
or even how those AI
like
works.
A grand algorithm.
Indeed. All right, let's see if we can get through one more round. Let's go back to Jason. There's another Tim now.
There's another Tim. Okay, TWiN, I couldn't open this paper directly, and it'll take me some time as my libraries are all closed until June. Hopefully they will reopen with public access to these articles. I did snoop and found a link to the commentary in Cell by Camila Belloni and Christian Lucia. I'll provide a link here. This show is a good follow to TWiN 15 with Helen Vuong. I especially enjoyed Tim's WAG speculations. You remember that, Tim?
No, not at all.
I'm like, what does that stand for?
Wild ass guesses.
Oh.
I'm not sure if you look back on these shows or not, but you should see all your faces light up when Tim takes one step after another off the deep end.
Tim's like, what is he talking about?
Or
perhaps he knows all too
make
well.
Me smile, crack my beer, and start to think as well as have a good time.
Good job.
There you go, Tim.
Oh, thanks, Tim.
Also,
thanks to you, Tim.
On microbiomes, but you might want to suggest some if you have for listeners to approach this work. You could sign off the week with the book you're reading currently or recently, any reading material accepted. I took the survey, but I've been thinking more about it. But maybe one thing you could do before you set off into your show is doing an icebreaker with an edge towards getting to know each other better, especially your guests. Twix does this with the weather reports. Twix?
Well, Twix is
biology.
Twix is just the name for all of the This Week in X, right?
Weather reports?
Yeah, we do that on
We
Twiv.
didn't do it today.
Yeah, we don't always do it here.
Very warm in
Hong
Hong
Kong.
Kong.
Many of these available. Aaron Welsh and Aaron Ullman Updike from this podcast will kill you. Talk about cocktails.
Oh, yeah, I've heard. Yeah, I know somebody. They talk about beers. That's their first one, first thing.
You all could do something else. Talk about a mental episode or disorder that you've experienced or with which you have come into contact. This neurological anecdote could be a case from Ori, a story from a colleague, a recent odd paper you read, neuralgia in your hands or a simple matter of deja vu. Just one account with a few lines of explanation could be enlightening, be relatable, and build interest in the show. Seth Grant has an anecdote that he has shared about how schizophrenia runs in his family that adds a ton of context to his work. So Seth Grant's a neuroscientist that works on synapses. talk about the most significant problems or least understood issues in neurology.
Again, none of us are neurologists, so.
You could alternate icebreaking. The victim of choice could suggest the question even. Topics like, where do emotions come from? What is a dream? Do we keep identity throughout our lives? What makes a sociopath? Are all day-to-day things that neurology can offer models and insights. Those are all good ideas. You could also talk about a hobby or something you're trying to learn. Your listeners are trying to learn science. They will relate. Share your guitar, beer making, daily life. People will find it funny. Keep it G-rated and suitably impersonal, but make sure it's authentic. Don't make anything up. We are all carrying around subject matter of this blog, regardless of whether we are thinking about it or not. Remind people how these shows relate to them right as they listen or eat their next meal. Thanks, Tim. Yeah, I mean, I think scientists, it's a good point. As scientists generally don't involve their own experiences and personalities in their science, or at least when they communicate to other scientists. And of course, we are human, and there really are stories behind a lot of the reasons for folks being scientists in the first place.
We are not talking to other scientists here. That's the thing. But I find, at least on TWiV, I talk about things that affect me, and it's very powerful. It resonates with people. They like to hear about not just the science, but also, hey, what's this issue I had? And we do that a lot on TWiV. I love it. I think it's great. And I can understand scientists are not used to that because it's not what they do. But as Tim says, it really engages people, especially if it's related to the subject matter of whatever podcast you're doing. So,
yeah, I mean, I coach this when we do the TED format talks, and academics sort of poo-poo the TED format largely because they bring these personal anecdotes into them to sort of gain trust with the audience and gain sort of a connection. But there's a reason why the TED format works in terms of being able to communicate ideas, and I think part of it is this relatability.
Yeah. Anyway, I encourage all of you to do this whenever you like. I mean, we tend to get in a rigid format and focus on the paper, but just if there's something that fits, just do it. I have no problem with that.
Should we do it now?
You can do it, Vivian. I think you might have some.
Yeah, I do. I do have some things. I have bipolar disorder. I also have a speech impediment, apparently. I have bipolar disorder and attention deficit disorder, not the hyperactive version, but the space cadet version. And I think that, this is a topic that I'm very interested in because I'm still, as I get older, and this is actually relates to one of the questions that we can get to in another episode about adolescence. And as you get older, you learn to reflect more on yourself, or in a different way, and gain more insight into your behaviors instead of just living deeply within them as you do when you're younger. And so I enjoy talking about bipolar disorder and ADD and ADHD because I'm still discovering things about how my brain works and how I have to adjust my world to fit within the larger structure that society expects us to all function in. So it has nothing to do, it doesn't have anything directly to do with my work, although I do study brain development. And I believe that these disorders, there's a kind of cluster of conditions, bipolar disorder, the attention deficit conditions, as well as autism, they all kind of hang out in the same space. And sometimes it's very difficult to tell which is at play or all three, and when do they rear their ugly heads, and when do they provide you advantage also. There's a big discussion in the, argument, fight, whatever you want to call it, in the ADD, ADHD community about people saying it's a superpower. And it's true in the sense that those of us who excel or sometimes struggle with it can reach levels of creativity and productivity that what are we saying? It's normal. It's the opposite of neurodiverse. It's like something. I don't know what the term is, but
neurotypical.
Neurotypical, right?
Yeah.
And that's true, but it is to the detriment of other things. So, for example, I can write an F32. It's a postdoctoral fellowship application. I can write it in 4 days and I will get the award, but that also means that I was absent from my family life for 4 days, and my daughter started having anxiety-induced, what's it called? Confused awakenings, which are like it's like the cousin of night terrors. So, and I think that there are a lot of people who are on the ADHD, bipolar, autism. I'll call it a spectrum, but spectrum kind of implies that there's two ends, and I don't believe that to be the case. I think so. There are probably a lot of people who are listening, or they know people who live with brains that work in the way that they do. And so I would love to talk more about that in the future, or do some papers that look at them.
Sure.
That's my shit.
Yeah, thanks for sharing. But wait, you can write an F32 in 4 days? I mean, a lot of us a month. We're absent from our families for a month.
No, yeah. So this is a true story, actually. I did write my F32 in 4 days, and
wow.
My mentor read it but made no comments, and actually said that it was the most pleasant and easiest grant writing experience he'd ever had. And then I submitted it, and I got it. So,
wow.
Cool.
But so the problem, but you said it takes a month. People who have ADD and ADHD suffer from something that's called time blindness, and so it's a kind of inability to gauge how long something is going to take, or how fast or slow time is passing. And
I think my partner has that.
Yeah. I mean, we're all kind of in there, right? But so if something is a month, my time horizon is really short. It's either today or not today, and so something that you're like, I'm going to work on this, so that I can get to my deadline. I'm just trying to figure out how one does that, because I just can't, for the life of me, figure out how to do that.
Do you think if you were to have taken 10 days to have written the grant?
Have bipolar in my family. I can't talk about it because, it's somebody else's medical information. But I'm coming to New Orleans, Vivian, in April. I'd love to have a coffee with you and talk about it.
Yeah, I would love to.
Because it's not being well managed, and it's a real problem. So
it's can be extremely disruptive, and they're all different flavors of it. Because all of these things are, nature and nurture. I think that there's there are definitely genetic predispositions, but as we talked about earlier, it's not like there's one gene that's going to cause this. But it's also the environment you grew up in. If you have a parent who has bipolar disorder, or, major depression or crippling anxiety, and you yourself have a genetic predisposition to this, that is, it's a dumpster fire at the end of the day. So, you know how to contact me. In what city I live. So, I would definitely love to get together.
I'll be, as I said, I'll be there in, I think it's April. So we'll connect. Vivian, can you take the next one, which I think is Jeff?
Sure. Hi, I've been a TWiV follower for some time, but just started listening to TWiN and was catching up. So this question is a couple of years out of date. It may even precede me joining. But maybe someone will have something useful to say about it anyway. I don't know. Useful is a strong word. In particular, regarding episode 2, those who fire together, wire together, TWF, TWT. That's a mouthful. There was a lot of great discussion of brain development, and the paper that was reviewed was over a decade old. But of course, as I'm sure you know, this theory is much older than that. In about the mid-1990s, there arose a fascinating twist on this thesis, primarily represented in Jeff Elman's book, Rethinking Innateness. Specifically, the hypothesis was that the timing of the development of different areas of the brain is organized such that the TWF, TWT, those that fire together, wire together model creates a hierarchy of areas representing at the early edges close to sensory inputs, lower. So then I think it's kind of, he's trying to show a progression here. Early edge, so it's close to sensory inputs, lower order representation, and progressing to the higher or farther from the sensory areas. So kind of like, I think what we call like secondary brain regions, where we're integrating information from multiple sources to multiple modalities. Elman and others demonstrated this effect in computational models, and these models actually became the basis for what we now call deep learning, which is the thing that makes most modern AI go. But the data at the time supporting this thesis came from very rough PET synchronization experiments. Don't ask me what PET stands for because I have forgotten. I'm sure somebody else here can tell you. I was wondering whether in the almost 30 years since this thesis was proposed, the basic hypothesis of what they called a wave of development has been observed, refined, rejected, forgotten. Here's a pointer to the paper which we originally proposed this model. And he also has the paper there that has the PET data.
Positron emission tomography.
Tomography.
Anybody have any insight on this?
I mean, in humans especially, we know that like the prefrontal cortex is the last part of the brain to develop, and it takes a long time. And even post-adolescence,
some of us are still working on it.
So for humans, I think it is true that there's it's a very protracted developmental window. Whereas the sensory modalities like the vision and hearing, those developmental windows are a lot more constrained and earlier. So I think that's held up. That these developmental windows, there are different time windows for different kinds of circuits in the brain, and it makes sense that you may want to start off with the basic sensory modalities and processes that you have to adapt to the world very quickly, otherwise you're going to die, versus higher cognitive function in humans. So I think that's panned out, and the sort of idea of developmental waves, I think it's definitely it's
paper in the show notes where so the original PET study that listener Jeff cited. So PET actually looks at glucose metabolism. So it's a way for you to kind of track the activity of the brain, roughly. And subsequently, so that paper was published a long time ago, 1987. And I dug around and I found a 2004 paper, which is still a long time ago, but a little bit more recent. And this paper looked at structural MRI. So it looks at the volume of the cortex and the gray matter density. And they found a similar kind of development unfolding from earlier senses, sensory, visual processing, and also somatosensory processing developing first, and then later developing are the so-called association cortices. So kind of prefrontal cortex and like maybe parietal lobe and maybe temporal lobe. So it seems to pan out. Yeah.
Yeah, because you can't know a priori what the mature brain is going to look like. So you have to start from the bottom. And then it's also, from when their conception, up until at some point, there are these extremely stereotyped processes that occur whether you're talking about brain development or you're talking about body patterning, like things have to occur in a certain sequence for the organism to even make it out into the outside world. And then, like Jason was saying, after that, you're either going to latch on, you're going to have the infrastructure to latch on to things that are important for your survival, or you're going to die. So, but then, yeah, where you're going to end up later, what the brain looks like in the adult is kind of an emergent property, which is, you know, it's interesting that we were talking earlier about trying to create a map or a connectome. I just find it hard to understand or imagine what that's going to look like for an adult brain, unless what the map really is showing is not literal connections of networks, but rather as a representation of potential for creating connections. If you see what I mean?
I think it's true that every single person's brain is going to be a different map. And so the question will come down to, are there principles of the map? Are there general sort of highways that could be, you know, common?
Mm-hmm.
All right, Tim, can you take the next one, please?
Sure. So Brenda writes, of interest, question mark. So she gave a link to a paper that and continues, at 74 years of age, I'm interested. So this is Brenda Steele from Blackhouse, Scotland. So the paper that Brenda was interested in, so I had a very quick look at it. The paper actually analyzed a large proteomic data sets that it seems to be collected for purposes that is not for Alzheimer's disease. But the researchers, you know, cleverly reanalyzed some of this data and found that there's some association between Alzheimer's disease. So they also have like patients' health status, and some of them happen to be Alzheimer's. Found an association between Alzheimer's disease and isomerization of aspartic acid in a peptide from tau. So that needs to be explained a little bit. So tau, we talked about earlier, is a protein that can become pathological in Alzheimer's disease. And it consists of amino acids, and one of the amino acids is aspartic acid. And apparently it can isomerize, which means it can go from kind of like if listeners took chemistry before, they remember this thing called chirality, where amino acid can be kind of in a right-hand form or in a left-hand form. And I guess this protein in Alzheimer's disease become more of the second different form than usual. And this usually happens when the protein has been sitting around for a long time, and weird chemical reaction can spontaneously occur. And so what this points to is actually that the neurons in Alzheimer's brain might be deficient in this garbage collection and disposal pathway called autophagy. So the paper suggests that perhaps Alzheimer's disease, this might be a indication that Alzheimer's disease may be linked with impaired autophagy. So which is, I think, very interesting concept. And autophagy is definitely a very, very important cellular process. And it's probably involved in quite a lot of diseases, actually, not just Alzheimer's. But that's the extent of my knowledge with autophagy, unfortunately.
Yeah, I mean, I think it's just one of the major ways that you degrade proteins, and actually autophagy, there's different kinds of autophagy where it also is important for degrading large structures like defective mitochondria and organelles in the cell. And it all comes down to this idea of proteostasis, where the cell needs to keep levels of proteins at a very constant state. And if you disrupt either side of things, if you make too much protein or if you degrade less protein, you're going to gunk up the system. And the problem is in neurons, because they age, they're as old as we are, It's just that these processes start to accumulate, and if you get a defect in any one of them, that can trigger, this pathology.
Jason, do you know anything about do neurons, can they put their trash out for microglia to engulf? Because there's quite a bit. So phagocytosis, or the eating of cells, of which there are different kinds depending on the situation, is my jam. But I've never thought about, I know there's a lot of research about microglial phagocytosis, or the ability for a microglial cell to engulf debris or dying cells, and how when that gets disrupted, or when there's defects in that it can lead to neurodegeneration and neuroinflammation. But do neurons put their trash out? Are they able to rely a little bit on these phagocytes to help reduce the burden of trash that they have, or stuff that they'd have to otherwise deal with themselves if their autophagy pathways aren't strong?
I think that there's an emerging idea that this is the case. That so, for example, we're studying these extracellular vesicles, which are packets of protein and RNA that get spat out by cells. And there is this idea that they could be another garbage disposal mechanism, and they get sent to microglia. And if that goes wrong, then you get accumulation of the junk. So, yeah, the role of microglia in Alzheimer's has been debated, whether it's good or bad.
It's both.
And, exactly. It's both. You need microglia to clean up the junk, but if they're overactivated, then they clean too much. That then they're going to start cleaning out things that you need.
All right, one more. This is from Ryan, who writes, "Dear TWiN, you have to look at this claim by Elon Musk." He sends a link to a article in The Hill called "Elon Musk Could Start Implanting Computer Chips in Human Brains this year," and this is dated February 2nd, 2021.
Yeah.
I don't know, and Ryan continues, "I don't know how true this claim is. There's a claim that Elon Musk is going to release a device where the chip will be implanted in brains. Initially, monkeys were used, but apparently it's in trials. Musk is working with the FDA." Anybody know anything about this?
Yeah. So he just had actually a expose, or not expose, but an update on this, and this is his company called Neuralink, and he's claiming that this chip that they can implant using a robot that they've developed, that they're going to try and get FDA approval for in the next 6 months. But after this whole sort of event that they held, there was a lot of outcry about whether they had been mistreating animals, and whether these animals could actually, whether the FDA would ever approve this, or at least certainly not in the next 6 months. But generally, the idea is that what they've done is used this brain-computer interface, and they've engineered this chip so that you can implant it in humans, and potentially, at least for things like amputees and prosthetics, that this could be a real advance. Partly because they've thrown a lot of money at sort of engineering the chip so that it works really well. But in terms of technology that they have actually developed, there's nothing that they've developed that's beyond what neuroscientists have been using in their labs. So I think that the key here is, can they actually engineer it to the point where it will be a very useful device for humans? And jury's a little out there, I think. But Tim, maybe you know more.
I remember reading, actually I'm on the Wikipedia site now. I think one of the aspirational goals for Neuralink is according to Elon Musk, he dreams of implanting electrodes so that he can save the brain state, a bit like a save game, so that sometime down the line, if something happened to your brain, you can just reload the game. So I don't know what something happened to your brain is. Is it just, if you had a stroke, which is a good idea, but I don't know how having electrodes is going to, promote your kind of bits of brain that is stroked out to come back. Right. Or is it just like you forgot?
Have you guys ever heard of altered carbon?
Yeah. I've read the book. Yeah.
I've read them too. It's a great show too, but this is very similar. Like you have a stack,
right.
which is like it's like a floppy disk. You on a floppy disk, and it inserts into the neck of essentially an like real live avatar.
I get asked this a lot, actually, and we're so far away from being able to download information from the brain that can recapitulate a brain that I'm skeptical it's going to happen in my lifetime that we'll even get to that point. To be honest,
isn't there a way to maybe I'm just making this up, but aren't there people or studies that have said they've tried to they're able to recreate an image of like a dream or something like that? Am I just making stuff up? Or?
I think I remember seeing this.
They could recreate. I think what they showed is they could recreate the visual image based on the visual activity that they're imaging in the brain.
Mm-hmm. Okay, so not quite, because they would provide the person with a stimulus.
Yeah.
And okay, so it's a little bit like,
and then match it.
You train the brain on a data set. Yeah. Okay, so not the same.
Yeah,
but I don't know about.
That's still a crude image. It's not like you could just recapitulate a whole dream experience.
Yeah, I just am not sure that that's just like too science fiction-y.
This is where Elon's he's not a scientist, he's a salesman to be honest. And so I feel bad for the scientists at Neuralink that are doing good work, that are trying to do things like solve manipulating prosthetics, or even being able to have people to walk. But then you've got Elon saying, oh yeah, we're going to download people's consciousness and then, solve everything.
Yeah, that just shows lack of understanding.
Well, he's clearly not understanding what went on in Wuhan and gain of function and all that based on his latest tweet, but that's another story.
Oh no.
How can one person do so much damage and also do some good at the same time, right?
Yeah.
It's like microglia.
Yeah. Anyway, that was, and it was a lot of fun. We have many more left. We'll tackle them another time. But that'll do it for TWiN 36. Show notes at microbe.tv/TWiN. If you have a question or a comment, you see now we're answering them, so please start sending them in. TWiN at microbe.tv, TWiN at microbe.tv. If you enjoy our work, consider supporting us. The parent company, Microbe TV, exists on your donations. It's a nonprofit in the U.S., and therefore anything you give to us is U.S. federal tax deductible. And you can go to microbe.tv/contribute. Many ways that you can contribute. Jason Shepard is at the University of Utah. He's Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent. Well, I guess maybe we're still going to have to migrate to another social media platform.
I don't know. The other one is not so great. No. For many reasons, so I'm just sticking on Twitter for now. Tim Chung is normally at New York University. Thanks, Tim.
Thanks, Vincent. Essentially, I might as well be on the moon with this lag.
It's not quite. It's not a second or so, but it's good. Tim, when are you coming back?
Around New Year.
I can hear my echo in his thing when he's ready to hear it. That's pretty funny. Vivian Marston's at Tulane. Thanks, Vivian.
Thank you so much, guys. This was so fun. I've been jonesing to do this for months.
We'll do it again. No problem.
Right.
By the way, all you guys should have received gift cards for Spike's T-shirts. I hope you got them. Yeah.
Mine is ordered and on the
I thought it
way.
was spam for a minute, and I was like, "What is this?" Oh, it's from Vincent.
It's not spam, but it's not spam, but it is unsolicited, so maybe that qualifies. But it's
it was great.
Free
I
T-shirt.
need
You
to
too, Tim.
order
Did
it.
you get your Spike shirt gift certificate?
Yep.
Okay, good. Get a Spike shirt. They're a lot of fun. The profits go to Microbe TV. I am. It's kind of patting myself on the
Paying
back.
yourself.
It's kind of weird, but that's the way it goes. But that's our last episode for 2022. You've been listening to This Week in Neuroscience. I forgot to tell you who I am. I'm Vincent Racaniello. I'm on Twitter at PROFVRR or at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next year.